Hi guys, welcome to the YouTube channel of Safe Paul Veterinary Services. Whenever we are going to talk about the bacterial diseases, their diagnosis and treatment, we must have a know-how about the differentiation of gram-positive and gram-negative bacteria. We all know about that gram-positive and gram-negative bacteria are differentiated basically due to the composition of their cell wall. If we are going to talk about the cell wall is composed of glycan backbone, mean a sugar that is made up of N-acetylmuramic acid and N-acetylglucosamine. They make the glycan backbone of the cell wall, and these glycan backbones are linked together with the short peptide chains of 4 to 5 amino acids. These are cross-linked. In this way, a cell wall is formed in its final composition. If we are going to talk about the difference between the cell wall of gram-positive and gram-negative bacteria, the gram-positive bacterial cell wall you can see here is made up of peptidoglycan. Just in the lower side you can see the cell membrane, and on the top side it is shown the peptidoglycan cell wall of the gram-positive bacteria. In the case of gram-negative bacteria, you can see here this plasma membrane, and in the middle the peptidoglycan, and in the outer layer again the lipid bilayer and some protein fragments in it. So there are two lipid bilayers and a peptidoglycan layer is sandwiched between them. That is the difference between gram-positive and gram-negative bacterial cell wall. If we are going to talk about the staining of gram-positive and gram-negative bacteria, as the gram-positive and gram-negative bacteria are differentiated on the basis of the gram stain, that has the four components: the primary stain, mordant, and decolorizing agent, and the final counter stain. The primary stain used in this staining is the crystal violet that gives purple color to the all the cells on which they are stained. And then is the mordant that intensifies the color of the primary stain. And then there is a decolorizer, and that in different cases, like in the gram negative case, it decolorizes the cell, and the cell then takes the color of the counter stain. Then the counter stain, that is saffron, that provides pink color to the decolorized cells. Here is the pictorial view. You can see here the primary stain is applied, and it gave purple color to all the cells. Then we have applied the grams iodine, which intensifies the color of the primary stain. And then we applied the 95% alcohol wash that has decolorized the gram negative cells. As we have seen earlier, that the gram negative cell is made up of a peptidoglycan layer that is sandwiched between uh, two lipid bilayers. In this case, alcohol disrupts the outer lipid bilayer, and then penetrates into peptidoglycan and disrupts the CV complex and dissolves it, and in this case decolorizes the gram negative cell. And again, if we apply this counter stain saffron, the gram negative cell takes that stain and are stained as pink color in the final slide. In the case of acid fast bacteria, we will discuss it in later. They are also like the gram positive bacteria, which have a peptidoglycan outer layer as a cell wall, and they have again mycolic acid in their cell wall that differentiates them from the gram positive bacteria. In the case of gram staining, they are stained like gram positive bacteria, but in the case of if we are going to differentiate them, then we use acid fast staining, in which they even resist the decolorization from the acid alcohol. Thank you very much.